I'm hoping that's worked. Can everybody see?
I can see it.
Yay!
So thanks for the opportunity to give you an overview of what we're proposing to do in the Ngāraki Taketake funded research area targeting Austropuccinia psidii effectors. So this project team is drawn together between Plant and Food, University of Canterbury and QDAF, Queensland Department of Agriculture and Fisheries. And it's largely going to be driven out of the University of Canterbury. Ren Dobson and Claudia Mesmera. But we now have in place a postdoc, Jenna Gykes, and an honours student, Tyler Johns, who will be working on this project for this academic year. Next slide, please, Anna. So a definition of effectors, they are relatively small proteins and they're expressed by plant pathogens to aid infection of specific plant species. So they tend to alter plant processes and drive the interaction between plants and their pathogens. Now we need to remember that we're dealing with a biotrophic pathogen in Austropuccinia. So that's a pathogen that keeps its host alive, doesn't kill it to reproduce, it keeps it alive. So one of the other roles of effectors in the biotrophic organism is to suppress the plant immune response and stop some of the plant host defense genes from killing the cell. So that's programmed cell death, which is one mechanism plants have to defend themselves, and animals have to defend themselves against pathogens. So they're very important in that whole infection process. Now we know that there are over 350 predicted effector genes in the genome. So that genome is available as a pre-publication, still available as a pre-publication paper on the G3 site. But again, they're predictions, but the programs that do those predictions are pretty good. Next slide, please, Anna. So effectors are only one type of protein that are expressed. So this diagram is taken from a review. It's now 2 years ago, I think it's 2019 review. So it's those orange colors are the effectors, and you can see that they come in a series of pulses. So you get bunches of or pulses of effectors during the infection process. That reddy color is all the other enzymes like cellulases, which cellulases of course digest plant cell walls, which are produced, and they're produced in general as a peak at the beginning and a peak at the end when the fungus is trying to burst out of the inside of the leaf or the tissue and sporulate and produce those spores. And the purple one is transporters. So that's things to move material in and out of the, particularly into the fungal pathogen whilst it's invading the tissues. So it's a very complex process, infection, and there's lots and lots of proteins interacting, being produced and interacting between the pathogen and the host at that time. Next slide, Anna, please. So I wanted to include this slide because why we propose to target effectors in the center there is because of a lot of the work that was done in some other programs to get us to the point to say we think we've got valid targets to go after in terms of trying to understand the very early stages of infection. So the first one is the Catalyst project, which I lead and is going to finish fairly shortly. And that was to screen New Zealand germplasm against the fungus and identify whether we did or didn't have resistance. And from that, we got phenotypic data, which showed that there was resistance, and it gave us material that we could begin to understand a little bit more about the infection process. That was also supported by extra funding from MPI and MPI 18608. And also that program gave us funding to get a pathogen genome, because without a genome, we do not have the resource to go and explore the genetic makeup of this pathogen. That ultimately, as most people know, led to that great trans-Tasman collaboration with the University of Sydney-led Australian group. And between us, we've got now a really good resource of a well-defined pathogen genome, which is being used both in work we're doing here and also in the ARC linkage, to which we're linked in terms of effectively exploring the basis of pathogenicity. Our focus is in Manuka. The Australians going to work on their species, and what we're hoping is we'll see commonality between the infection process of the fungus on the different species. This is at the molecular level, which means that targets we identify in one species should be suitable for targeting in other species. That ARC linkage got a bit hung up in the COVID and ARC processes, and it's just getting going now. So we're sort of slightly ahead of our Australian colleagues right now. And then in the Beyond Myrtle Rust, the Endeavour funded program, is where we did our initial transcript analysis. So that was taking those phenotypes, those plants that we knew were resistant or susceptible, and looking at how they were responding at an RNA level to being infected, and also what genes the pathogen was expressing. And we could take all of that data and put it together as a proposal for NRT to look at these 3 effectors in one enzyme. And the broad outline of this program is to produce and purify the proteins. They've got that skill set at the University of Canterbury at the Biomolecular Interaction Centre. And Jenna did her PhD on producing and purifying difficult proteins, identify their probable tertiary structure, and identify the plant proteins that the effectors target. And we'll be doing that hopefully by being able to generate a yeast 2 hybrid library and find the targets for those effectors in the Manuka genome. And from that, we would like to be able to propose some strategies for how we might target those effectors to stop them doing their job. So a fairly complex slide, but it's a fairly complicated background to where we are right now. So the next slide, please, Anna. So just wanted to, so this is one of the key results from the first transcriptome experiment. We're currently trying to set up another one to validate this and extend it. So 1, 6, and 5, so those green colored circles. So this is a differential expression analysis. So we're looking at the time phase. So we're looking at that, the pulses, effectively, how the amount of these proteins is shifting over time. So we've got those 3 effectors, and we've also identified this cellulase. Now they've got strong green, most of them have got strong green lines linking them. That means they're in positive correlation, and they're a positive, good, strong correlation. So that means they're all fluxing or changing at the same time. So they're all going up and they're all going down at the same time. And that the numbers actually reflect the order they came out on the differential expression. So that's over time. So you might say, well, 2 is higher in that list, but you'll notice that 2 is mainly linked to the other 5 by red lines, which means it's fluxing in the opposite way to the effectors. So for that reason, even though it's high on the list, it's not as good a target, or not a target that we're as interested in terms of going after an early stage proteins. 4 on that list is actually completely unknown. So when you challenge the databases around the world with the protein that's 4, it comes up with no hits. So we also don't want to use 4 because we don't know what it is. We have no idea what it's doing. So we've got 3 effectors and a cellulase as our initial targets, and they're the targets we're taking forward into the project. I think, is that the last? That might be the last slide, I think. Anna? Yep. Yep. So hopefully I've done my best to explain what we're driving at in this project. Thanks.
Kia ora. Thank you. And we do have plenty of time for questions. And I'm just looking if there's any in the chat. There's none in the chat. But if anybody has a question, just raise your hand or start speaking.
I'd like to ask a question of Grant. Rob Beresford here.
Hey, Rob.
Hi, Grant. So I just wanted to be sure which host genotype are you using for these studies? Are you using several host genotypes? And it is Manuka, is that correct?
It's Manuka. Yeah, so it's the host genotypes that we've identified in the screening at QDAF. So there's a mixture of leaf, both types of leaf resistances, so the immune and the hypersensitive, and as well as susceptibles. And we're still just trying to finalize what the yeast to hybrid library will look like, but it's probably going to be a combination of those mixtures.
Right. So that's all based on seedling reaction types, is it?
It's all based on seedling reactions. Yep.
Do you have any sort of information about how that changes as the plants age and grow? Because there is the observation from the field that Manuka doesn't show rust infection or very little as older plants, yet the seedlings can be quite susceptible.
Well, I think it's a case of, yeah, at the end of the day, that pathology assay is absolutely optimized for the pathogen. And so in that respect, it's a very tough assay on the plants. But those plants have been maintained right now at QDAF and been cut back and then reflushed, and they show the same phenotype so far. And I guess you'd say how far they're probably still an early flush, even though they're a couple of years old, Rob. So no, we don't know how well that translates through into an aged plant at this point.
Yeah. Oh well, I guess that's just something to bear in mind for the future.
It's one of the things to bear in mind for this work, Rob.
Thanks, Grant. That's good.
So.
Kia ora. We had a question from Karen van der Woud. Is swamp Maori included in your study?
No, largely because they've been such a problem to germinate the seeds. So we have taken seed over to, I mean, that's more the Catalyst program, but we have taken seed over there. They do have some swamp Maori, but I'm not sure whether they've got enough. I haven't talked to Louise for a while to know whether there's enough plants there to do a statistically relevant inoculation. It's been a real problem getting the seeds to germinate.
I have a question. So with this knowledge, how does it help us manage the pathogen and the disease?
Well, right now we've got, from my perspective, we've got one management tool. That's to spray prophylactically with a fairly nasty fungicide. This at least allows us to, if we get to where we want to get to, we have an understanding of some of the key fungal proteins that are driving the initial part of the infection process, establishment process. Can they become targets for some novel intervention strategy? And I have no idea what that novel intervention strategy will look like until such time as we've got some structures and things to work with. But we can't go around, I mean, Angus established fairly early on that you could protect plants in the bush by spraying them with fungicide, and as soon as you stopped, they got infected. But that's just not an option. So do we have another mechanism that we can use? Well, this is one of many, hopefully, potential paths that people are following up. Ours, it's going to be fairly molecular, but what that final tool will look like will not be molecular. It'll be trying to target those proteins.
Goodbye. Just checking in, any other questions? No, thank you. So thanks, Grant, for this. It's really good that we have this opportunity to hear a bit more in detail about what work is going on and to ask each other questions. So if anything else comes up later, don't hesitate to follow up with Grant or via Ina through the NRT email as well.
Excuse Katrin.
Yeah.
It's Simon Cook here. How are you going?
Good. You show up as Anna as well.
I think I'm called Anna. Yeah. I'm much better at Anna anyway. So, hey, can I just ask a question? And will the effectors will they target Myrtle specific proteins, or are they targeting proteins that are obviously found in many other families?
Well, based on all the work that's been done on effectors over the years, you know, especially a lot of that work that was done by the Canberra group, there's a lot of very specific interactions going on between a rust pathogen and its host. So that work in Canberra was done, of course, on rust fungi, but we do not, given that Austropuccinia only infects myrtaceous plants, we believe that they're interacting with myrtaceous proteins. How different those proteins look from the plant proteins of other species, we won't know until we can identify the targets, Simon. But I suspect there'll be those subtle differences between the proteins in wheat, barley, oats, etc., and myrtaceous that allow the differentiation of those pathogens to their hosts. So I think they'll be myrtaceous specific, but what we would hopefully that they will be widespread through the, it is they are key proteins in the initial infection process. So if you can somehow stop those proteins interacting with the effectors, then you might have something that can be applied across many myrtaceae